Good afternoon, in some cases probably also good evening from my side. I'm currently dialing from Switzerland, more precisely from the Swiss Alps, where I'm spending the weekend with my son. It is also a pleasure and an honor to be here for the first time presenting our NTSAD conference. And I do hope I can briefly present to you what is the project and the scientific hypothesis behind GAIN Therapeutics. To start with, I don't certainly have to explain the enzyme involved in deficiency and finally leading to the development of the disease, which is beta-galactosidase. And certainly I don't have to speak in front of families and experts about the high unmet medical need that still are present into this patient population. The only thing I would like to underline is that brain is an important aspect of these diseases. And the other aspect I also would like to mention that the same enzyme is not only involved in GM1 gangliosidosis, but it is also involved in Morquio B, which is a related disease, which has more cartilage and bone feature. Most probably you know very well the various tools that are or potentially could be available to treat lysosomal enzymes. And there are esteemed colleagues from other companies having some other approaches. Our approach, as you can see into the middle of the slide, it is to try to stimulate a more, let's call it, physiological activity of the enzyme. So what is the scientific hypothesis behind us that we are able to enhance beta-gal into the cells, and that this approach will finally lead to a substrate depletion, which is at the root cause of the molecular onset of the disease. What is also something we want to achieve, and we have other programs ongoing, such as in Gaucher, soon in Krabbe, and so on. Our target is to have oral medication that are available and can be distributed into hard to treat tissues such as brain, bone, cartilage, and other type of tissue. Very briefly on the science, and Sue was telling me not to go probably too much into details, but what I would like to say is to the audience, which is a mixed audience, protein, when they are built in a specific compartment of the cell, which is called the endoplasmic reticulum. Within this, let's say, complex, there is a system which is called protein quality control. You don't have to be a scientist, but this protein quality control is basically assessing the right three-dimensional conformational structure of the disease. I don't know if you can see my cursor on the slides, otherwise on the left side, what I'm saying is if you have an enzyme or a protein which is not properly formed, the protein quality control will process it and destroy it because it is considered as a faulty protein. When that is happening, the protein cannot reach the final cellular localization. You probably know that the important cellular localization for GM1, like for Morquio B and other lysosomal disorder, are the lysosome. So it means that genetic mutation leads to an impaired three-dimensional structure. The protein is discarded by the protein quality control, cannot reach the lysosome, and within the lysosome, that, as you know, they are a bit the recycle bin of our cells, substrates, they cannot be processed, and so they start to build up. The substrate buildup is connected in several of the diseases to degenerative diseases, so cell death. And this is the hypothesis we are working on, and I will briefly explain to you. So what we are trying to do, and here you see a little star, we gave star to our molecules that are otherwise scientifically called structurally targeted allosteric regulator. We want to stabilize a faulty protein. Why we want to stabilize a faulty protein? Because we want the faulty protein to have a better shape, escape protein quality control, and be able to travel through the cells to reach the lysosome. Because if you have enough quantity of the enzyme, you will most probably decrease the substrate, which in GM1 gangliosidosis is GM1 ganglioside. So this is what we want to work on. Very briefly, again, it's a scientific slide, but in order to do so, you have two possibilities. An enzyme has, and here is depicted in red, what is called an active site. So it's the site of the protein which is responsible for chopping down or for metabolizing the substrate. And this site is normally scientifically easy to recognize, easy to target, easy to find drugs. The problem is that if you are occupying that site, you are also in competition with the substrate. So it means you can stabilize, you can reach the lysosome, but you are occupying the site, and so you cannot process the substrate. We took a different approach. We want to use other points on the surface of protein that when you can develop drugs, they lead to a better structure of the enzyme, therefore avoiding protein quality control, therefore having more protein into the lysosome. How do we do that? I will certainly not enter what we do. What I can just say is that to do so, we need three things. We need a published three-dimensional structure. We need supercomputer. There are about 500 worldwide, and so they are not so difficult to find. And we need algorithm that can specifically study which part of the protein, if we would be targeting it, will lead to a stabilization and therefore more abundant present in the cellular localization. And this is what again we have. We have a proprietary algorithm that I'm not explaining to you, which is assessing only those sites, and normally it is 1 or 2 per protein that can stabilize the protein. All that is done in silico, and also the initial compound selection, it's all done in silico, and you can identify a sites in about 2-3 weeks. I don't go through the whole cascade, but after we applied and we studied the three-dimensional structure, we started to have about 700 compounds that we have selected. Some they were commercially available, other we generated ourselves. We went through a screening cascade, which is basically assessing various properties of a molecule to become a drug. As of today, and after a couple of years of work, we have one molecule. And I will briefly explain to you what this molecule is and where do we stand. As right now, we are implementing 2 animal model study, one in Morquio B and the other in GM1 gangliosidosis. The compound has a feature for which it is specifically binding to beta-gal. It is specifically binding in a dose-dependent manner. Why that is important? Because as such, you can adapt the dosage to the final administration to the various patients. Something important for us was to test, and this is just a very high-level summary, the molecules in patient-derived cells having a Morquio B or GM1 gangliosidosis mutation, and with our main compound and the backup compound, we do see enzyme enhancement, which make us progressing the project further. I was telling you about that a faulty protein cannot reach the lysosome, or can reduce, or the presence can be very much reduced. What we have done, and unfortunately this is not a picture because those they are not quantitative, but very simply what we have done, and it is in the blue, and this is both in the wild type, so the normal enzyme, in the Morquio B and the GM1, we assess, or we try to reach, if we are able to enhance the lysosomal fraction. So the promise was, are we able to get more protein into the lysosome? And this assay, which is a lysosomal fraction assay, is indicated that in all 3 settings, the 2 genetic one and in the wild type, we are reaching and getting more abundant protein into the lysosome. What we have done, and this is just an initial data, it's not the final one that we are currently doing. We looked at fibroblasts that were accumulating GM1 gangliosides. So those are two different experiments, just to guide you through. The green is a fluorescently marked GM1 ganglioside, so the substrate that, if accumulated, is leading to the disease. The blue is a dye for the nuclei, just to identify each single cell. And what you can see is that when we apply one of the compound, one of the most advanced compound, you do see that the green fluorescence is remarkably diminishing, and therefore we are stimulating substrate depletion or toxic clearance. Obviously, those were initial data. We wanted to assess if the molecule has some features to become drugs. So we've been performing studies to see if the drug, the molecule, could be orally bioavailable. And as you can see, both in plasma and in brain, the molecule GT513 can not only reach the brain, but can reach the brain also as an oral medication. We were looking, because of the two diseases, at brain and bone, if we were able, with various dosages, if we were able or not to reach the bone and the brain. And this is actually what we are seeing. In all three dosages that we tested, we can reach the brain and the bone, and the effect is over time. We have been performing, and we are producing more, acute toxicity study. So the molecule, it is safe. And where do we stand right now? We took the decision to advance the program, and we started, for the family, probably is a technical term. We started with synthesis scale up. What does it mean? Instead of producing grams, now we are producing kilos. And the reason is to produce the kilos is because we will start at the end of this year all the regulatory process to finally ask at the end of 2002 an IND, which is an application to start clinical trial for the drug. So this molecule has, again, all the feature. Why as the feature? We saw gene-specific enzyme enhancement. We have seen substrate depletion. We have seen stabilization of the enzyme and protein maturation, which means going into the lysosome. We have a molecule which goes into the brain, it goes into the bone, it is orally bioavailable, and in terms of tolerability and safety, looks not to have red flags. We have to await the regulatory process, but so far it looks good. So therefore, we took the decision to start the IND regulatory process at the end of this year. It was for us important to have access to various tools and to various inputs. The list is probably not complete, but there are various stakeholder, medical doctors, patient association help us to get access to patient-derived cells, to animal models, and I would like to thank them all, and also reminding that the project has been supported by a European Union grant. And finally, we are still a very small company. Basically, this is the whole company is working on. In a way, we are also a big family, and therefore we take family needs and patient needs very seriously and very personally. Thank you for your attention.
Thank you, Dr. Bellotto. That was fantastic. Just to clarify, because I may have been distracted and looking something else, you plan to submit the IND when?
End of 2022.
Okay, end of 2022. And you're beginning interactions with the FDA at the end of this year. Is that right?
Second half, yes.
Second half. Fantastic.